Hello, this is Jason,
And Michaela.
and today we're going to discuss the results of our work as the FPLC team.
Start off, what is MGH? MGH is a protein. The M stands for malate dehydrogenase, which is an enzyme used in the citric acid cycle. The G is for the green fluorescent protein that it has, which gives it the observable neon green color. The H is for the 6 histidines in a row, which makes it unique and what helps us separate protein.
We begin our experiment with cells previously introduced to the MGH plasmid. These transformed cells, the ones who took up the plasmid, will demonstrate a green color on an ampicillin plate. A single green colony was taken and inoculated in broth, and this broth was left to grow until it reached an optical density of at 600 nanometers of 0.60.
Once our broth contains sufficient amount of cell growth, we then need them to overproduce our MGH protein. To accomplish this, we added IPTG, which serves as a chemical signal for cells to produce MGH by translating our plasmid. Typically, cells will try to avoid having too many foreign proteins by producing inclusion bodies. To avoid this, we incubate the cells in the fridge, which reduces inclusion bodies against MGH, but conversely takes more time to produce. At this point, MGH is abundant but stuck inside living cells.
Two methods were employed to disintegrate the cellular membrane, or lyse the cells, to release MGH into solution. Firstly, lysozymes are added to break glycosidic linkages, a component of the bacterial cell wall. Then, sonication is used to disrupt and destroy weakened cell membranes with intense sound waves. The resultant solution, known as cleared lysate, is a low-salt buffer that has two components added to it. One is DTT, which essentially prevents formation of disulfide bridges between unwanted and wanted proteins, and the other is PMFS, a bacterial protease inhibitor that prevents breakdown of our protein. At this stage, the clear lysate, containing all the proteins in solution, is ready to be purified.
Use FPLC to separate the proteins we want from the ones we don't in our cleared lysate. This is a preparatory technique as it involves our entire experiment up until this point. We use affinity chromatography because it's very efficient, a one-and-done purification. NINTA resin takes advantage of the His6 tag on MGH protein, allowing for us to narrow our FPLC purification process to His tag proteins, a somewhat uncommon attribute.
The buffer we used had to be compatible with all resin choices. We couldn't use EDTA because it would remove nickel from the column. For the lysis buffer, there is low salt and no imidazole, so there is no selective precipitation of proteins. This is loaded at 0 minutes. The loading buffer, buffer A, has a high salt and low imidazole. For the first 30 minutes, the contaminant proteins are rinsed through. Finally, the elution buffer, buffer B, comes in around 31 to 70 minutes and has a high salt and high imidazole concentration. The high imidazole allows for competition for the His tag spot, with the imidazole taking over the spot on the column. And basically this completes the purification.
This figure shows the result of the aforementioned purification scheme. We can see the solution conductivity based on salt concentration, pictured in the red, absorbance at 280, which is the wavelength for protein absorbance, pictured in blue, and the fraction number in green on the bottom. There is a clear spike in absorbance at 280 at 0 to 30 minutes during the loading buffer A stage. This most likely represents unwanted proteins. A sharp decrease in conductivity is also seen as large amounts of protein passing through the meter will interfere with salt concentration. Further on the right of the trace, around 50 to 70 minutes, there is an increase in absorbance at 280. This is most likely His6 tag protein eluted from buffer B that has high imidazole to take the His tag proteins off the nickel column. There is no change in conductivity as there are not enough proteins to make a significant difference to salt concentration.
Thinking about the conclusion here, for the total protein concentration, which is related to the absorbance at 280, from our FPLC trace, we can conclusively say that there are considerable proteins that should be examined at fractions 2 through 9 and 17 through 23. These are the values that demonstrated the highest absorbance at 280 nanometers, and the subsequent protein concentrations, since this is the absorption range for proteins. Since we don't know which peak is necessarily MGH, it's probably the best idea to test both of these peaks just to make sure we don't miss anything.
Typically, there is the highest likelihood of MGH with his 6 tag being in fractions 17 to 23. This is when buffer B is introduced with high imidazole that kicks MGH off the nickel column. In addition to this, the fractions appeared slightly green colored, which indicates the presence of MGH.
In conclusion, fractions 2 through 9 and 17 through 23 should be taken for further spectroscopic analysis. These are the fractions that represented the highest A to E and subsequent protein concentrations. Any spike in protein concentration is worthwhile to examine. However, we suggest that fractions 17 through 23 should be taken into careful consideration, as the nature of our FPLC and buffer setup predict that his tag proteins like MGH will be eluded later in the fractions. In addition to this, it appears that fractions 17 through 23 are green in color, which could point out MGH. Overall, fractions 2 through 9 as well as 17 through 23 will be sent to the spectroscopy team for analysis, with the general sense that MGH is present in